Fonzo, who will be presenting his recent work, Corticolimbic Mechanisms of Affective Disorder Vulnerability, Expression, and Treatment. Many of you know Greg, and some of you may recognize him because he was a joint doctoral program student here, working with Murray and with Martin, and then went away to Memphis for a clinical psychology internship, and then has been at Stanford for his postdoctoral work. Greg submitted a K award that got reviewed quite favorably, which is very, very exciting, and we would love to talk more about Greg coming here to San Diego, to the VA, and to the Department of Psychiatry. So we're excited to hear about your recent work, Greg.
Excellent. Thank you so much. I'm mic'd already? Okay. Excellent.
Sorry.
There you go, no problem. Yeah, and so thank you, Susan, for that introduction. And thank you, everybody, for being here today. I'm excited to share with you some of the work I've been doing over the years. And I'm actually going to begin kind of at the start and then kind of take you through just a bird's eye view of the kind of the trajectory of my research over the years, looking at corticolimbic mechanisms underlying affective disorder, vulnerability, expression, and treatment. And so I thought I might start at the end, kind of, in terms of, so this is currently where I'm sort of looking forward to in terms of the vision I have at the current point for where I see my career heading and sort of the big picture. And then I'll take you through the steps I kind of went through over the years in order to get to this point. So ultimately, kind of my grand scientific vision, if I ever get to achieve it, will be to study individuals with affective symptoms. So looking at this in a transdiagnostic sense, exaggerated negative affect and diminished positive affect, and using neuroscience tools in order to understand the brain basis of some of these symptoms, as well as characterizing behavior. And then from that, use advanced statistics and computational models in order to begin to construct models of information processing in the brain that underlie these aspects of symptomatology. From there, I want to understand how our existing efficacious treatments impact these processes in the brain in order to result in a resolution of symptoms, both in terms of mechanisms in the brain as well as phenotypic characteristics at baseline that predispose individuals to having a more favorable treatment response. And from there, we kind of have two separate lines. One is use predictive biomarkers in order to parse heterogeneity of individuals at baseline and be able to establish predictive biomarkers, so essentially the characteristics that will predispose individuals to responding favorably to a given treatment. On the other hand, I want to use some of the mechanistic understanding gained from this work in order to be able to identify targets for improving treatment outcomes in these individuals. And from there, use that in order to design novel treatments that engage these targets, assess the target engagement and subsequent symptom change in an iterative process, and then eventually establish efficacy of these new treatments. This is kind of the grand vision, and I'm going to tell you a little bit how I kind of got to this point, and hopefully that will start to make some sense. So in the beginning, there was graduate school, and that was here in San Diego, which was a great place to do my graduate work. And I came here and worked, as Susan said, with Murray Steen and Martin Paulus. And my early work focused on understanding negative valence systems in anxiety and post-traumatic stress disorders. And so one construct that's often used to study negative valence systems is threat reactivity, and which we know is definitely relevant to the clinical symptomatology we see in these disorders in terms of being an increased reactivity to threatening stimuli. And so this is typically studied in the laboratory by presenting a stimulus such as a fearful face. And in the brain, this evokes a response in several regions, primary of which would be the amygdala, which is a collection of subcortical gray matter nuclei located in the medial temporal lobe. It's critically involved in sensing or detecting a salient environmental stimulus and then engaging subsequent responses throughout the brain to deal with that. Further up in the processing stream, we have the insular cortex, which is a structure that we now know is critically involved in a construct known as interoception, or the sense of the overall physiological condition of the body. And as such, it's critically important in representing the physiological states that underlie our most subjective emotional experiences. And then in the prefrontal cortex, we have an area of the brain in the medial prefrontal cortex known as the dorsal anterior cingulate and the dorsomedial prefrontal cortex, which is involved in some of the secondary attentional modulation that occurs when a threatening stimulus is detected. And so from this, we can say that threat reactivity is a process that's distributed both cortically and subcortically throughout the brain. Now, when I first came to San Diego, let me tell you a little bit first about the processes involved in regulating this threat reactivity. So I'm going to describe two primary emotional regulatory processes, the first of which is an implicit regulation process, This is something that occurs automatically, and it's thought not to need any conscious or deliberate effort on the individual's part to regulate emotion. And it's thought to be situated in the ventromedial prefrontal cortex. The second broad category is the explicit regulation system, and this is involved in effortful strategies to regulate one's emotional state. The typical being cognitive reappraisal. We often teach people in psychotherapies. It's secondary and deliberate and involves areas such as the dorsal anterior cingulate, as well as regions of the lateral prefrontal cortex, such as the dorsolateral PFC, which is involved in higher order cognitive functions in humans, as well as areas of the ventrolateral prefrontal cortex, which are involved in processes such as inhibition and stimulus-driven attentional orienting. So coming to San Diego, I began working with Murray and Martin, trying to understand some of the neurocircuitry underlying these negative valence systems in anxiety disorders. And at the time, there was a strong movement towards moving away from DSM categories per se, and looking at things more in terms of dimensional constructs that might underlie various overlapping symptom structures that we see across these diagnoses. And as such, we had a study ongoing at the time that was recruiting individuals with three primary different anxiety disorders: panic disorder, social anxiety disorder, generalized anxiety disorder, and we had healthy controls as well for comparison purposes. Now, the goal here was to begin to understand what are some of the characteristics in the brain that relate across these disorder categories, as well as potentially might distinguish them. And so we used a common threat reactivity paradigm at the time, which basically involved an implicit emotional face matching task. And so the goal of the participant was to match the affect of the top face to one of the two faces on the bottom. There was also a shape matching control condition involved as well to control for the behavioral process of processing. And so looking at this task and then contrasting trials of matching to fearful versus happy faces, which were two of the primary emotion conditions on this paradigm, we observed that there was a common hyperactivity in the amygdala to fear processing. And as you can see here on this graph, the contrast of matching to fearful versus happy engaged the amygdala more so across all three anxiety diagnoses relative to healthy controls. So the shaded blue bars are the fear responses, and then the three on the left are the anxiety disorders, and the healthy controls are on the right. Now, we thought this was interesting because this is the first time we had detected, or at least compared across these three anxiety disorders and observed a similar abnormality. And we wanted to go one step further in kind of pushing from DSM more towards RDoC, and we asked the question whether this might be representative of a continuous affective trait that is perhaps overrepresented in these categories. And so what we did is we used a voxel-wise mapping approach, and we compared a continuous measure of negative affectivity, trait anxiety in this case, and examined how that related to brain activation during this contrast. And indeed, by doing that, we observed that a nearly overlapping region of the right amygdala was also significantly related, trait anxiety was significantly related to activation in this region. So from this, we can make the statement perhaps that this amygdalar hyperactivity we see across anxiety disorders is both a categorical abnormality as well as representing a continuous affective trait. Now, this amygdalar hyperactivity is something that was very well known at the time to be characteristic of anxiety disorders as well as post-traumatic stress disorder. And at the time, it was well understood that if you show an individual with PTSD an empty threat stimulus like a fearful face, that they would display an exaggerated amygdala response. However, we know that this threat reactivity system is distributed throughout the brain. And so we began to hone in on another key player in this threat reactivity circuit, which is the insula, and questioning whether we would see similar abnormalities in post-traumatic stress disorder. And so we had a study ongoing at the time that was looking at PTSD due to intimate partner violence, and we recruited women with this condition as well as 12 healthy controls, and we administered the same face processing paradigm that we used in the prior study as well. And the interest here was in establishing if the insula does indeed show an abnormal response to these empty threat cues as we see in the amygdala. And indeed, we observed first a hyperactive response in the amygdala to these empty threat cues, which gives us some indication that the paradigm was valid for replicating that phenotype. And indeed, when examining the anterior insula there on the right side, you can see that individuals with PTSD also displayed an elevated abnormal response of the insula in response to threat processing. So from this, we can say that this general fear abnormality that we see in post-traumatic stress disorder is something that's distributed across this threat processing system. Now, what about the prefrontal cortex? So I had mentioned that that is involved as well in threat reactivity. And at the time, it wasn't really well understood what the different roles of these regions were. And so we reasoned that the prefrontal cortex might be involved in processing aspects of the threatening stimulus that are more related to higher order or contextual characteristics, as opposed to being things that are just the affect of the face per se. So we wanted to capitalize on the homogeneity of this sample, and basically all were women who experienced or got post-traumatic stress disorder through exposure to intimate partner violence at the hands of a male intimate partner. So we reasoned that that homogeneity could be exploited here for examining how, for example, the processing of male facial characteristics relative to female facial characteristics might actually induce a threat reactivity response in a separate region of the brain. And the important thing to note here is that the emotions were counterbalanced across male and female faces, so this contrast doesn't actually have an emotional component to it, just the face gender. And indeed, we observed that when women processed these faces to male versus female targets, that we selectively induced abnormalities in the medial prefrontal cortex. Specifically, women with intimate partner violence, post-traumatic stress disorder, displayed an elevated response of the dorsal anterior cingulate to processing male versus female faces. And thus, from this, we can surmise that different aspects of stimulus characteristics can dissociate cortical and limbic threat reactivity abnormalities. So to quickly summarize, I've shown you that anxiety disorders and PTSD are characterized both by common increased threat reactivity of negative valence brain systems, and this manifests as distributed corticolimbic abnormalities. And at this point in my career, I began to start thinking about overlapping symptoms across diagnoses as being characterized by a shared circuit dysfunction, and this really got me into thinking about things in terms of more of a neurobiological sense as opposed to a diagnostic one. In terms of development as a scientist at this point in my career, I picked up a lot of skills actually, acquisition, analysis, and interpretation of fMRI data, recruitment, retention, and assessment of clinical populations for research, and learning a lot about the functional neuroanatomy of threat, fear, and anxiety. Now, at this point in sort of my work and my research trajectory, I've been doing a lot of these case control comparisons and seeing these kind of blobs of activation on the brain that indicate some difference for one group relative to another during some behavioral paradigm. But I became interested in wondering if we could bring a greater level of inference to understanding what these activation differences might mean. And so I reasoned that if we begin to look at risk factors and vulnerability factors that might relate certain individual characteristics to an abnormal phenotype, that might begin to give us a better level of inference in understanding what these abnormalities actually mean. So take, for example, a person that we might see in the clinic who has a hyperactive amygdala response. Now, typically when we look at these case control comparisons, we tend to assume that this hyperactive amygdala response that we see is different from healthy controls is a consequence of disorder development, here depicted in that red blowout on the continuum there. But in actuality, it's entirely possible and perhaps more interesting that this might represent a pre-existing vulnerability factor that predisposes individuals to developing the disorder. Now, typically, this isn't something that we can state with any certainty, but I began to think about how we might be able to get at this question. And it occurred to me that one way to do that would be to start to look at the phenotype in the adult and examine the relationship of certain characteristics to known risk factors. And so, for this purpose, I looked at childhood maltreatment as a potent risk factor that predisposes individuals to developing anxiety disorders later in life. And so childhood maltreatment typically involves the experiences of emotional abuse, emotional neglect, physical abuse, physical neglect, and sexual abuse, oftentimes that co-occur together in the same individual, usually at the hands of a caregiver. And over the course of development, predisposes individuals to a host of negative mental health outcomes, including anxiety disorders, depression, post-traumatic stress disorder, substance use disorders, and personality disorders. And so the question I wanted to address is, if we understand something about a person's profile in terms of their exposure to maltreatment, can we use that information to begin to identify potential mechanisms or patterns in the brain that might relate childhood maltreatment to the later expression of symptoms? And in this process, I really wanted to address using this approach if childhood maltreatment history could be used to understand or hypothesize or identify a candidate mechanism that might relate to the relationship. And so I began to recruit and aggregate individuals from ongoing studies at the time, ended up with about 180 individuals with post-traumatic stress disorder, anxiety, and depressive disorders. And the goal here was to use a voxel-level mediation modeling approach in order to address the question of what areas in the brain, in terms of brain activity and functional dynamics, mediated the relationship between childhood maltreatment history and anxiety symptoms in adulthood. And so, of course, having to using this threat reactivity paradigm, we had to look in the amygdala, and indeed we observed that amygdalar activation to processing fear versus the shape matching condition did indeed mediate this relationship, such that greater levels of activation were associated or mediated the relationship between childhood maltreatment and anxiety symptoms, thus explaining some of the variance there. And this is a mediation plot, and on the x-axis we have brain activation, on the y-axis we have our anxiety measure, which was the brief symptom inventory anxiety subscale, and then those three lines indicate the relationship between activation and anxiety symptoms at the different levels of childhood maltreatment, with the center line being the mean level of maltreatment across the sample, and the line above and below being one standard deviation above and below. And so the greater the spread of those lines, the larger the mediation effect. And indeed, we observed, looking at another emotion contrast of anger versus shapes, a similar mediation relationship in the left amygdala this time, wherein greater activation mediated that relationship. Now, from this pattern, we might surmise that there's some increased emotional reactivity process in the brain that might account for this vulnerability effect of childhood maltreatment leading to the later development of anxiety symptoms. And so we might also then surmise that there might be a breakdown in prefrontal structures involved in regulating this response. And indeed, when we looked at the prefrontal cortex, we saw effects in the right dorsolateral PFC for fear versus shape processing. And indeed, that was in opposite relationship to that observed in the amygdala. So decreasing activation in the structure mediated the relationship between maltreatment and anxiety symptoms in adulthood. And likewise, when looking at anger, we observed a very similar effect as well in the right dorsolateral prefrontal cortex. So to quickly summarize, this is one way, or this exemplifies how certain risk factors can shape brain processes in order to support disorder emergence. Now, it's important to note here that this is entirely cross-sectional, but it does give us a candidate mechanism that we might target with future longitudinal studies. And specifically, at least childhood maltreatment seems to lead to the later development of anxiety, potentially by disrupting the balance of threat reactivity and regulatory processes in the brain. And in terms of my development at this point, this got me moving away from just strictly cross-sectional comparisons and brain blobs to thinking about using risk profiling and statistical modeling in order to be able to identify and hypothesize candidate mechanisms that might relate these processes over time. And in terms of my development as a scientist, I just began to learn how to test mechanistic hypotheses that related some of these processes, as well as applying these models to large neuroimaging data sets. So after graduate school, I moved to Stanford, and this is where I did a lot of my more recent work. And at this point, I became very interested in using neuroscience tools such as imaging and TMS in order to be able to understand how to inform clinical care, or basically how that might work. And so currently, our treatment armamentarium for these disorders includes a combination of one or both of these treatments, being psychotherapy and medications. So if two individuals walk into the clinic, let's say Joe and Jane, we have no idea really who is likely to respond to which treatment. And so it becomes a game of trial and error in order to determine, what is going to be the most effective treatment for this individual. Now, ideally, we'd like to be able to use some sort of targeted brain and behavioral assessment in order to be able to identify the characteristics of the individual that will best match them with the most appropriate treatment. Additionally, we can use this information in order to be able to understand mechanisms underlying existing treatments and then use that to develop novel treatments. So really, that's the picture I think that the field is moving towards. And so using this sort of framework, we came at this first from studying PTSD psychotherapy. And so this was a randomized clinical trial for prolonged exposure treatment, which basically used prolonged exposure, first off, is a very highly standardized and efficacious treatment for treating PTSD. So it does give us that advantage from a research perspective of being able to isolate effects easily. This was a RCT with imaging, both at baseline and following 1 month after treatment. Individuals also received the same imaging battery. And we randomized 36 individuals with PTSD to immediate treatment and 30 individuals to a treatment waitlist, at which point, after the waitlist, they also received the same efficacious treatment. We used intent to treat analyses with linear mixed models in order to maintain that randomization, both on the voxel by voxel level with imaging, as well as on clinical outcomes. Now, the primary hypothesis underlying this study was that psychotherapy works by improving one's ability to regulate emotional responses to reactive, or to stimuli that would cause a reactive emotional response. So as such, we had a battery of tasks that assessed these processes. The first was an emotional reactivity paradigm, which assessed threat reactivity, and basically just used fearful and neutral faces presented in a color tint and asked the individual to identify the color tint of the face and ignoring the emotional expression. So as such, it's a measure of implicit emotional reactivity. The second paradigm we use is a kind of quite popular emotional conflict task, which the way it works is the individual is asked to identify the facial affect of the face and ignore the overlaid emotion word. And so there were fearful and happy faces, each with an overlaid emotion word, either fear or happy. Now, when the word is congruent with the face, then it's quite easy to identify the facial affect. But when the word is incongruent, that causes a conflict effect. Actually, individuals slow down on their reaction time to identifying the facial affect due to the word. Now, the interesting thing about this paradigm is that when a conflict trial is preceded by another conflict trial, there's actually an adaptation effect that occurs, wherein on the second conflict trial, individuals actually mitigate their reaction time slowdown, and they're more quick to identify the facial affect. So there's some adaptation effect that occurs from conflict trial to conflict trial. And this has been identified to relate to a specific process in the brain as well, wherein on the second conflict trial, individuals increase activation in the ventromedial prefrontal cortex and decrease activation in conflict responsive structures in the dorsolateral and ventrolateral prefrontal cortices. And this has been touted as a measure of implicit emotional regulatory capacity. So that was a measure of implicit emotion regulation, and we also had a measure of explicit emotion regulation, or cognitive reappraisal, in which the individual was prompted at the beginning of the trial to either decrease their emotional responses to a subsequent negative picture, or to just look at the negative picture and feel their emotional response. They then looked at the picture for about 4 to 6 seconds and then gave a rating regarding their level of negative affect at that time. So let's begin with looking at the baseline characteristics that moderated responses to prolonged exposure. And so beginning with the emotional reactivity task, and knowing that the emotional reactivity is a process that's distributed throughout the brain, we indeed observed that there were effects at baseline that moderated subsequent responses to prolonged exposure. So looking at the prefrontal cortex first, we can see that we found moderating effects in areas of both the dorsal anterior cingulate as well as bilaterally in the dorsolateral prefrontal cortices. Now, when you examine these effects, the way to read these graphs are we have the prolonged exposure group in the left-hand quadrant, and then the weightless group on the right, and then pre and post scores on the CAPS, which is the clinician-administered PTSD scale on the y-axis. And the solid line is individuals who had a above median level of activation in that region, and the dotted line is individuals who did not activate as much to that contrast. And what we can see is that individuals who showed greater activation at baseline in those regions showed a steeper drop in symptoms for prolonged exposure relative to waitlist. So this is an effect that was specific to prolonged exposure and was different in waitlist, so it's a moderation effect. Now likewise, we also observed a moderation effect in the amygdala. And actually this one was in the opposite direction. So individuals who displayed less amygdala responses to these threat cues actually displayed a more favorable response to subsequent prolonged exposure. So from this, we can now begin to form a picture that there is perhaps a phenotype at baseline that will allow individuals to respond more favorably to this treatment. And this phenotype seems to involve a greater prefrontal deployment and less amygdala recruitment when processing these threat reactivity responses. Now, it's perhaps tempting in order to interpret this as being a prefrontal inhibition of the amygdala. And that's oftentimes what we might like to tell ourselves in terms of the story here. But however, we don't really know for sure if that's actually what's going on due to the inherently correlative nature of fMRI. We'd like to assume that that's an inhibition effect, but we don't know for sure, even though the pattern would suggest so. However, we wanted to bring another level of causal inference to these data, and so we utilized the tool at Stanford, which is concurrent transcranial magnetic stimulation and fMRI. So TMS is a tool, transcranial magnetic stimulation, I call it TMS for short, is a tool that allows us to selectively activate particular regions of the brain using a magnetic coil that we place on the scalp, and by turning that on, we can then selectively activate a region of the brain located directly below that coil. And we combine this with imaging, interleaving TMS and fMRI, and we were able to image the downstream effects of modulating a particular brain region. And we had about half of our sample that were randomized to immediate treatment undergo this TMS fMRI. And in order to probe this potential circuit relationship here between the right dorsolateral prefrontal cortex and the amygdala, we looked at stimulation of a region we'll call the posterior middle frontal gyrus. This is an area of the right dorsolateral prefrontal cortex that is part of the resting state executive control network, and which overlapped with our moderation effect we detected during the emotional reactivity paradigm. So we thought this might be a good site to begin to establish a causal probe of that relationship. And as a control site, we used another area of the right dorsolateral prefrontal cortex, which we'll call the anterior middle frontal gyrus. It was actually part of a totally different resting state network, being the salience network, and it did not overlap at all with any of the task moderation effects. So we expected that that site would not be tapping the similar process. What we did is we examined the effect of stimulating these regions on the rest of the brain, and specifically honed in on the amygdala, the response that we saw to moderate responses during emotional reactivity. And then we extracted the causal modulation from these regions and tested it to see whether it would moderate treatment response. And indeed, we observed that for individuals who showed a greater downregulation of the amygdala due to right posterior middle frontal gyrus TMS relative to the anterior middle frontal gyrus, which showed no effect actually, they tended to do better on subsequent prolonged exposure treatment. So from this, we now have, this is, I think, a really cool example of how we can sort of start to use some of these neuroscience tools in order to be able to understand causal relationships in the brain, and specifically how those causal relationships might impact one's ability to respond to a subsequent treatment. Now, turning to the regulatory paradigms we looked at, I'll just sort of break the news and say that we did not detect any moderation effects during the explicit emotion regulation paradigm. However, we did in fact detect a moderation effect during the implicit emotional regulatory task that we use, which is that emotional conflict paradigm. And this was as expected in the ventromedial prefrontal cortex. So at baseline, individuals who were better able to bring on that region when adapting to emotional conflict tended to show a more favorable response to prolonged exposure treatment. So we can, from this, state that the greater one's ability to bring the ventromedial prefrontal cortex online during an implicit emotion regulation paradigm, the better your subsequent response to an exposure-based treatment for PTSD. So to quickly summarize, these corticolimbic activation dynamics actually moderate PTSD psychotherapy outcome. And specifically, this is characterized by greater prefrontal control over the amygdala threat reactivity response, tends to predict a more favorable response to treatment, as well as one's ability to deploy the ventromedial prefrontal cortex when implicitly regulating emotional salience. And I think the interesting thing here is that these effects were detected only when individuals were not deliberately regulating their emotional state, which actually jives really well with the clinical delivery of the treatment, in which it's heavily emphasized to individuals that they are supposed to feel their emotional responses and not deliberately try to regulate them. So I think essentially what we're detecting here is a circuit correlate of one's ability to maintain engagement with those emotional responses and then bring on ventromedial prefrontal regions in order to habituate to the emotional salience. Now, as a development as a scientist at this point, I was able to deliver psychotherapy in this trial, so that was an interesting experience for me, learning how to do that. Also, I learned proper statistical analysis of randomized clinical trials using linear mixed models and integrating that with imaging, and getting a good training in transcranial magnetic stimulation and how to integrate that with fMRI. Now, what about the changes following prolonged exposure? So the predominant view in the field nowadays in terms of how psychotherapy works is by basically downregulating limbic responses and increasing people's ability to use their prefrontal cortex or to bring that online to regulate their emotional state. So given that framework, we might expect to see changes in the brain during emotional reactivity or threat reactivity. Unfortunately, we did not see any changes that were significantly different between prolonged exposure and waitlist anywhere in the brain during emotional reactivity. So we cannot provide any support for that aspect of the theory. Now, we might also suspect that perhaps the moderation effect we detected during implicit emotion regulation might also be something that changes and perhaps is boosted following treatment. However, again, we observed no significant effects or no significant changes between prolonged exposure and waitlist anywhere in the brain and during that particular behavioral process. We did, however, detect one change in one particular brain region during one behavioral paradigm, and that was during this explicit emotion regulation paradigm. So when individuals were deliberately trying to downregulate their affective responses, they brought online an area of the brain that was not otherwise active at pre-treatment. Now, this area of the brain is known as the lateral frontopolar cortex. So this is part of Brodmann Area 10, for any of you out there who know Brodmann areas, which is actually a very unique cytoarchitectonic region of the brain. It's one of the most highly evolved in humans and actually lacks a homologue in higher primates. It also tends to be exclusively interconnected with higher order associative regions of the brain, and it basically doesn't have any connections to sensory motor regions. So it seems to be involved basically in some of the higher order information processing, at least from its connections. And it does tend to segregate with coactivation analyses and resting state connectivity with more posterior areas of the dorsolateral prefrontal cortex involved in executive control and higher order cognitive functions. So we might say that psychotherapy, at least from this data, seems to enhance frontal recruitment during explicit attempts to regulate one's emotional state. And that is at least the predominant change that we observed in this sample. Now, to better characterize this frontopolar effect and what it might mean, we looked at connectivity changes following treatment. So we extracted signal from that region and then ran a connectivity analysis on the individual level to see how the coordinated function of that region might change over the course of treatment. And used intent to treat analyses and basically observed that there was a change in one brain region as well, and this involved a selective increase in connectivity, which wasn't present at pre-treatment with the ventromedial prefrontal cortex. And as such, that is a key site that we know involved in implicit emotional regulation. And so given the fact that the frontopolar cortex is part of the explicit emotional regulatory circuit, and we're seeing increases in connectivity with the regions of the brain involved in implicitly regulating emotional state, we can perhaps postulate that psychotherapy integrates explicit and implicit systems in the frontal lobes in order to be able to perhaps regulate emotions more effectively. Moreover, I just wanted to point out that this frontopolar connectivity change is something that was clinically relevant as well. So the greater one's ability to, or the greater the increases in activation in that region following treatment, the greater the decreases we see in certain PTSD symptoms, specifically hyperarousal. And we also see a similar relationship with self-reported psychological well-being, which is a subscale from the WHO quality of life inventory. And individuals who more greatly increase activation in that region tend to show greater improvements in quality of psychological well-being as well. Okay, so to summarize this data, psychotherapy, at least in this sample and in these analyses, appears to work primarily by shifting frontal lobe function, which to me was kind of surprising. I would not have hypothesized that a priori. I was expecting to see more emotional limbic changes. And additionally, this is only when deliberately regulating one's affective state, which stands in nice contrast to what we actually observe to moderate treatment responses. So this begins to give us a picture now that the things that actually moderate or predict who will respond to a treatment may not actually be the things that will change with treatment, and that there might be secondary processes that are engaged over time that actually serve to maintain the beneficial effects of that intervention. And so the way I'm thinking about it now is that psychotherapy seems to work in improving outcomes for people in terms of reducing symptoms and improving quality of life by providing an enhanced mode of being able to explicitly regulate your emotional state. And that enhanced mode might be a greater integration of various emotion regulation systems within the frontal lobes. So I'm going to shift gears now a little bit and talk about the other primary arm of treatment that we use for treating affective disorders, and would be antidepressants. And here I'm going to focus specifically on major depressive disorder, for which these medications were first developed. Now, this data is actually brand new. It hasn't been presented anywhere yet, so I'm excited to kind of share it with you guys. So we know that antidepressant treatments are an effective treatment specifically for major depressive disorder as well as anxiety disorders and to some extent PTSD. They're widely utilized. However, we know that as well that not every individual is going to respond to an antidepressant the same. So there's a lot of variability there. About 50% of individuals will require a second form of antidepressant treatment, and only about 30 to 50% of those individuals will actually reach symptom remission. So thus there's a lot of improvement here that can be made, and I would argue that improving the clinical outcomes with antidepressants involves a targeted treatment selection. So knowing who's likely to respond to which antidepressant and being able to funnel individuals to the correct one. So a study that was designed to address this very question is known as the EMBARC Study, and stands for Establishing Moderators and Biosignatures of Antidepressant Responses for Clinical Care. So this is really the largest randomized clinical trial looking at biological measures to predict antidepressant responses. It's a multi-site initiative, I believe 4 sites altogether, looking at early onset recurrent MDD, randomizing individuals to sertraline or placebo, about 120 to 130 in each arm across 4 different sites. And they received imaging measures both at baseline and 1 week after starting the medications. And today I'm going to focus on the baseline phenotype that predicts a favorable response to sertraline, which was the antidepressant assessed in this study, and specifically using this emotional conflict test that we saw to also show a moderation signal for psychotherapy in PTSD. And so looking at these conflict regulation dynamics, specifically this conflict regulation contrast, we see very prominent effects across the whole brain for a moderation response. So this is FDR corrected across the entire brain. So it actually surprised me how robust this signal is. And we see it in the areas that we know are conflict responsive, including, kind of hard to see, I'll just state it, but the dorsal anterior cingulate, the dorsolateral prefrontal cortices, and the anterior insula. Now, in order to begin to understand what these effects mean, we started to extract signal from these regions and examine how they related to antidepressant responses. What we see is that across areas of the canonical conflict responsive network, including the dorsal anterior cingulate, the frontopolar cortex, the lateral portion, and the anterior insula, we see if we split the sample on the median level of activation just to visualize these effects, that individuals who more effectively dampen responses in these regions during conflict adaptation show a much steeper drop in symptoms. So the graphs on the left-hand side are individuals who more effectively dampen responses in these regions, and the graph on the right is individuals who did not as effectively dampen responses. And you can see that the treatment effect, which is HAM-D 17 symptoms on the y-axis, that we see a differentiation only for those who are able to regulate their conflict responsive networks. Moreover, when you look at remission rates, you can see that for sertraline versus placebo, we now have quite a large difference in remission rates, whereas before, just looking across the entire sample, there actually was no statistically significant effect of sertraline on treating MDD symptoms across the entire sample without examining these moderators. So this is kind of exciting. So we have now a biological signal that will predict who is going to respond to antidepressants. Now we wanted to take this a step further and see how useful this could be on the individual level for predicting responses to treatment. And so what we did is we used a parcellation of the brain. This is from Schaefer et al. 2017, which is about 250 parcels across the entire brain. And we extracted average activation for each individual for each parcel. And we used a machine learning approach called sparse Bayesian learning in order to identify the multivariate pattern across these various areas that would be able to best predict treatment change on the individual level. And indeed, we observed that while looking at the sparse weights, which are basically the weighting of these various regions to predicting response, we can see that they're widely distributed throughout the brain, including some of those conflict responsive regions we showed earlier. And then we used the tenfold cross-validation in order to see how well we could predict on the individual level. And by doing this, we can see here that on the x-axis we have true change in HAM-D pre minus post, and on the y-axis we have the change predicted by the model. And there's a significant relationship there, and moreover, it captures about 33% of the variance in treatment response. So this is really kind of cool. And then when we basically took the same classifier to see if it would work in placebo, to see if it wasn't just sort of a placebo response, we see that there's actually no relationship there at all. So this classifier seems to be specific to sertraline itself and isn't something that we see as just part of the general placebo effect. To quickly summarize these findings, the brain's capacity to adaptively regulate conflict-responsive regions moderates subsequent antidepressant treatment response, specifically for sertraline. And basically, the more effective or the better able you are to dampen responses in those regions, the better your response to subsequent sertraline treatment. And this is really exciting, I think, because now we have an example of how biology can inform treatment suitability in mental health, which is, I think, really cool. So now I'm going to talk a little bit about moving into the great unknown, which will be kind of moving forward into the future. And so one thing I wanted to point out is that thinking about how to kind of take this forward into an independent research trajectory, I started to think about some of the things I noticed in our PTSD psychotherapy sample. So both during the moderation effect we saw during emotional conflict regulation, as well as the connectivity increase we saw with the frontopolar cortex during cognitive reappraisal, you can see prominent effects in the ventral striatum for both. And so this interested me because neither of these paradigms really taps any sort of reward or positive valence, which is what the ventral striatum is classically associated with, but yet we're seeing very strong signal there. So I began to think about, why don't we study positive affect in PTSD more frequently? Fear and threat are definitely clinically prominent and things that we see quite often, but we also know that diminished positive affect is a prominent component of the symptomatology in PTSD. And I think it's been this way for many years, that the study of trauma psychopathology really has a very heavy emphasis on the fear-based symptoms, perhaps to the detriment of the diminished positive affect that we see in the disorder, including things such as the anhedonia, emotional numbing, diminished interest, and depressed mood that often characterizes these individuals. Now, I would argue that these are clinically relevant symptoms. It's not that we don't study them because they're not important. Individuals with greater levels of diminished positive affect display worse outcomes to our existing evidence-based treatments. They display a more chronic course of the disorder, as well as increased levels of suicidality and greater levels of functional impairment. So these are actually very clinically relevant, and I think a very important target for us to begin to probe more systematically. And I would argue that trauma science is really desperately in need of a brain-based characterization of the diminished positive affect that we see following trauma. So positive affect is typically probed in the laboratory using reward paradigms. So rewards basically engage the substrates that are known to underlie positive affect. These include areas such as the ventromedial prefrontal cortex, the nucleus accumbens and ventral striatum, as well as the midbrain ventral tegmental area. Now, reward processing also benefits from a really well-developed construct that allows us to begin to parse apart different components of information processing involved in the behavioral process of making decisions in order to accumulate rewards. And that also really interested me as a way to start to expand this line of research. So this construct is known as reinforcement learning. And essentially, reinforcement learning is a type of machine learning wherein mathematical equations are used to model the information processing underlying an agent's actions in some environment in order to accumulate rewards. That's kind of the textbook definition. Now, the way it works is we would, for example, measure the brain using something like imaging, and as well as behavioral responses during some reward processing task or reinforcement learning paradigm. And then use computational modeling and advanced statistics in order to be able to construct a model of how the different components of reward information are represented and processed in the brain during this behavior on a trial-to-trial basis. And thus we can then begin to relate dynamics of the brain itself during this behavior to aspects of information processing, which gives us I think a much stronger inferential lens with which to view some of these brain activation findings. So reinforcement learning is, just to briefly illustrate how this works. So let's say we have an agent in some environment, and he's trying to accumulate rewards, as any agent should. And so he has two doors, door A and door B, and he's going to, through trial and error, learn which door he needs to go through in order to receive the best possible rewards. So first he chooses door A, opens it up, and lo and behold, receives a large reward. So he's also going to try door B, naturally. And when he opens door B, he only receives a much smaller reward. So in this case, learning begins to take place, and he begins to understand that door A is associated with a much larger likelihood of a future reward. Now, the components involved in processing and learning this information tend to be instantiated in distinct nodes of the reward circuit. So, for example, door A we know is associated, at least from the prior example, with a likelihood of a larger future reward. And as such, we can say that door A has a large value associated with it, meaning that individuals, when they see that door A, they know that a large reward is likely to occur. That tends to engage a region of the brain called the ventromedial prefrontal cortex. So that is, we can say the VMPFC is responsive to value. On the other hand, door B, which has a smaller reward associated with it, also has a smaller value signal associated with it, and also tends to induce much less of a response in the ventromedial prefrontal cortex. So thus we can say the ventromedial prefrontal cortex, in this context, appears to encode value. Now what about when outcomes are different than expected? So, for example, if we have this individual open door A and receive a small reward, which is not what he expected, now he is receiving an outcome that is different than what was expected, in this case worse than expected. This is called a prediction error. And this is what's used to drive learning. It's basically through trial and error, learning which, when outcomes are different than expected, being able to update your value signals in order to effectively learn stimulus-reward contingencies. And when an individual has a negative prediction error, which would be when outcomes are worse than expected, it tends to drive down signal in areas of the brain such as the ventral striatum and the ventral tegmental area, which are classically dopaminergically innervated, and dopamine is thought to really compute these prediction errors. Now, on the other hand, if this individual opens door B and receives a large reward, this is an outcome better than expected. In this case, this tends to drive up signal in the ventral striatum and the ventral tegmental area. So we can see that these regions encode a signed prediction error signal. So using this framework of reinforcement learning and beginning to think about PTSD potentially and the reward abnormalities associated with it, I began to kind of probe the circuit using data that we had at the time. And so what I did is I took the congruent happy and fear trials from that emotional conflict task in our PTSD psychotherapy sample, and I contrasted them just to see what the positive valence-related signal might be. And indeed, we did observe activation in our healthy controls in the canonical reward circuit during this contrast, so it suggests that at least that the happy faces are engaging those structures as we might expect. What we did observe, however, was PTSD displayed abnormalities in this process, specifically decreased or hypoactivation of the ventromedial prefrontal cortex, and interestingly increased or hyperactivation of the ventral striatum and the ventral tegmental area. So now we're beginning to see that PTSD has some sense of an abnormal reward circuit response, at least to these ostensibly rewarding stimulus cues. Now, interestingly, these are actually clinically significant in that the greater the level of hyperactivation in the ventral striatum, the greater the level of anhedonia in the individuals with PTSD. Moreover, the greater the deficiency in activation in the ventromedial prefrontal cortex, the greater the levels of numbing symptoms of post-traumatic stress disorder. So this is now beginning to give us some picture that this reward circuit dynamics have some perhaps relationship to symptom expression, and thus might be a clinically relevant circuit to go after. Now, I wanted to examine more closely whether this circuit was operating as an integrated entity, and so I became interested in a technique at the time known as dynamic causal modeling, which is a way to measure information transfer within a closed system. And so the idea here is that we take the signal in these three regions of the reward circuit, the VMPFC, ventral striatum, and VTA, and we begin by modeling intrinsic connectivity between them, or intrinsic connections. And these are based on sort of existing theory regarding tracer studies that relate these circuits to one another. And then we see how a rewarding stimulus perturbs the connections in this system. So in this case, it would be the happy faces from the emotional conflict task, which enters the system through the ventromedial prefrontal cortex. And this is all using like Bayesian statistics and stuff in order to define the best fitting model. And indeed, we found that this rewarding stimulus did modulate all of the connections of the circuit. So it suggests that this is operating as an integrated reward circuit in the context of this paradigm. And when we compared the modulatory relationships between individuals with PTSD and trauma-exposed healthy controls, what we observed was a selective deficiency in directed connectivity between the ventral tegmental area to the ventral striatum, as well as back from the ventral striatum to the ventral tegmental area in individuals with PTSD relative to healthy controls. So thus, these individuals have what we might say regionally exaggerated responses in those regions, in that they're deploying greater resources there, but there's less of a coherent information transfer between them. Suggests a kind of a loud but noisy signaling within that area of the reward circuit. Now, I wanted to take this a step further to see if we could identify a similar signal using computational measures of reinforcement learning. Now, this is in a completely different sample of individuals. We had a behavioral paradigm that was a sort of a reward learning paradigm, and it was looked at this in veterans with and without PTSD. And the way this worked is they were presented with some ambiguous stimulus and asked whether the stimulus would predict sun or rain. And each stimulus would predict one of the two 80% of the time and the other one 20% of the time. So it was a probabilistic sort of trade-off there. Now, when individuals chose correctly, they received a reward, and when individuals chose the incorrect option, they got no feedback, so essentially nothing. So what I did is I took the choice behavior from these individuals in terms of decisions, reaction times, and used a Q-learning computational model in order to get at the reinforcement learning parameters of value and prediction errors relating to reward predicting stimuli. And indeed, we observed that PTSD showed a selective deficit in being able to maintain value signals during this paradigm, and indeed they showed an elevated positive prediction error relative to trauma-exposed healthy controls. So this actually provides a very nice scaffolding with the imaging data within which we're seeing diminished ventromedial prefrontal cortex activity, which does tend to compute value signals, and exaggerated ventrostriatal and ventral tegmental area activity, which tends to be the area in the brain involved in encoding prediction errors. So from this, we can say that PTSD displays, at least preliminarily, an abnormal reward circuit response, as well as abnormal reward information processing, and these abnormalities are related to aspects of diminished positive affect. And so this is really was kind of the springboard for getting the K23 funded. So in the proximal future, the research focus is going to be on executing on this K23 on reward in post-trauma psychopathology, which I propose to use a transdiagnostic conceptualization there, so I don't have to necessarily tie myself into the diagnostic categories that seem to be changing over the years very frequently. And so really the goal here is to recruit 45 individuals with a post-trauma psychopathology, in this case diminished positive affect, and 45 trauma-exposed healthy controls, and really characterize the phenotype in terms of understanding how their brain processes rewards differently, as well as how their reward behavior, or the behavior to accumulate rewards, might be abnormal. Then use various computational models and statistical methods in order to be able to construct models of information flow in the brain involved in representing this reward information. And then finally relate that to aspects of symptoms and functional impairment to begin to be able to understand how some of this behavior and information processing might relate to specific symptoms. In terms of the training component, it's going to allow me to get training in reward processing paradigms, theory, and neurocircuitry, some training in basic and translational neuroscience, understanding how to build and construct and test computational models of information processing, starting first with reinforcement learning and then expanding to others, and then how to integrate these computationally derived information components with imaging in order to be able to track how the brain processes this information. And then on the latter end, also build computational models of brain function itself to see how circuits relate, or how nodes communicate within a circuit. And then finally, more general grant writing, professional development, and clinical research skills. So I've been told it's important to kind of express like what's on your T-shirt in these talks, so I figured I would just kind of throw it out there as a, at least my idea at this point, what I think it's going to be. And so I really want to be the post-trauma computational neuropsychiatry guy. That would be my goal, I think, if at the end of the K, I'm kind of known as that. And that involves elements of imaging, reward behavior, computational psychiatry, and then building brain information processing models, as well as using that to understand mechanisms of symptoms. And then moving forward from that, I want to take this T-shirt and incorporate it into a larger T-shirt, wherein I start to look at things in a computationally informed trauma experimental medicine approach. And the idea here would be to understand how existing treatments impact the signals that we detected in the beginning phases in terms of computational constructs involved in reward processing and diminished positive affect, use that to develop targets to understand what changes in the brain and how that impacts symptoms, and then potentially engage these targets with novel treatments if they're not adequately engaged by existing treatment armamentarium. So briefly, I want to quickly thank my mentors over the years. Of course, Murray Steen and Martin Paulus, who are my grad school mentors, and Alan Simmons as well, who served as a grad school mentor, and then my current postdoctoral mentor, Amit Etkin. Thank you very much, and I'll be happy to answer any questions.
Questions, comments?
Yeah, try.
The mic.
Well, I thought really interesting and comprehensive talk, and I'm not sure how long you've been gone for, but you've been very productive since leaving, so hopefully we weren't holding you back here. So I'm really, I think it's great that you're sort of branching out and studying the positive valence system and reward processing, which obviously plays a big role in PTSD and related anxiety and depressive disorders. So sort of thinking about some of your data, so first of all, I was struck by the increased activation in the ventral striatum, getting very preliminary data in response to congruent happy faces. And I'm wondering, how much of that do you think is sort of driven by the contrast? So you're contrasting against fear-related stimuli, again, a trauma sample, and then sort of maybe a bigger question, which is, in these sorts of reward processing paradigms, do you think it's important to disentangle the sort of negative valence or threat or punishment processing from reward processing? Do you separate them out into separate tasks, or how do you grapple with that? Because again, a lot of the reward paradigms, there's a cost potentially of not getting the reward or of getting punished.
Right. Those are excellent questions. Yeah, so actually you brought up things that actually went into my revisions for the K. So essentially it was... Yeah, thank you. So the exaggerated ventral striatal response was also surprising to me. I was not quite sure what to make of that. There's a couple of ways I could potentially explain it, but these are just hypotheses at this point. One is that essentially the ventral striatum, if we're detecting a dopaminergic signal, we know that is heavily involved in wanting an incentive salience more so than the actual experience of liking or experiencing a reward. So there could be a dissociation there between the ability to want something versus the ability to experience the pleasure from getting that reward. The other thought that occurred to me is that since it's involved in prediction errors, that we have to take the context of the task into account as well. And in this paradigm, we do have quick trials of fearful as well as happy faces presented in succession. So this could be a function of the fact that context is also saturated to some extent by threatening cues. And that actually was an important thing that made me begin to separate out in the K. I have basically reward-only runs of these tasks as well as reward combined with punishment. And the idea there is to develop a pure reward probe as well as see how that interacts with potentially negative stimuli.
So of kind of your findings so far, have you incorporated thoughts into how you'd want to modify a therapy for somebody who'd been in a traumatic experience and is having symptoms?
Yeah, it's a great question. So I think I was talking about this with Alan just a little while ago. But actually, one thing that I find really exciting from a conceptual perspective is the development of MDMA-assisted psychotherapy for PTSD. So that is a very interesting thing from the construct of the sense that we have no other treatments for PTSD, at least currently established, that work by not directly targeting the fear-based symptoms and trying to induce learning in that respect. So it's an interesting idea to use something like MDMA, which we know is a potent sort of boosting of the reward system, and that, moreover, that actually seems to work for individuals that don't respond to these other treatments. So that it's an interesting pharmacological manipulation of the circuit, I think, for sure. There's, of course, other issues surrounding that, in terms of legality and potential side effects. But it does give an indication that perhaps of a proof of concept, maybe, that this might be something that could have legs in the future for improving treatments. The other thought I had in regards to how to improve positive affect might be taking some of the interventions that we know work in, for example, depression, like behavioral activation and pleasant activity scheduling, and things that we don't typically use for trauma treatments, but then somehow making that an adjunctive component to it. It's potentially, I think the idea would be to really balance reward and engaging the fear circuit in order to produce new learning that would occur, and finding the optimal way to do that I think is really the target.
Great. Is there an MDMA clinic at the VA yet?
They're working on ketamine. That's a couple years out. Any come after the marijuana clinic?
Yeah.
Are you also going to try to connect this to like blood biomarkers or like other biomarkers that you can find other than like the biosignature?
Yeah, no, that's a great question. Yes, I would like to. I think it's a matter of if I had the funds to collect that data. But I think imaging is a cool tool. I mean, we can really get a good coverage of the brain with it, but at the same time, it's a very hard thing to translate into clinical practice, because we can't get people to just like go get an fMRI scan. Yeah, it's not cost effective. So I think there's a couple of routes for that. One is using EEG as a more clinic-friendly tool and potentially translating fMRI signal into EEG signal in order to be able to kind of use and translate some of these findings into clinic-friendly kind of measures. The other is, as you mentioned, using other sorts of predictive biomarker approaches like blood, cortisol, genetics, for example, immune responses, inflammatory responses. And my sense is that none in and of itself is going to be very useful. I think we're going to have to use something like that sparse Bayesian learning in order to combine across multiple measures and find the best combination of predictors.
Well, there may be other questions, but you have a busy schedule yet.
Yes.
You still have 2 interviews and a dinner, so hopefully you can you have the resilience to hang in there. But we do have a small tradition here that if we enjoy the talk, you get a small reward.
Excellent.
I don't know which region is activated as a result. Yeah, there you go. But welcome back to the Department of Psychiatry at
Thank you.
UCSD. Wear it with pride.
Excellent.
And hopefully you'll bring it here in the future.
Thank you.
So thanks very much.
I appreciate it. Thank you so much.